Bismillahirrahmanirrahim. Assalamu alaikum. In this video we will study about the synthesis of the AMP and GMP from the IMP. This is a two-step energy and nitrogen requiring process. The energy from this step is derived from the ATP in case of the formation of GTP, and the energy for the formation of the ATP is derived from the GTP. The nitrogen required for the synthesis of the AMP is derived from the aspartate, whereas the nitrogen that is required in the formation of the GMP is derived from the glutamine. Now let's see these steps that lead to the formation of AMP, GMP, and then we will further study about the formation of GTP and ATP from these two. So if we see the synthesis of the ATP first, then we can see that in the first step the IMP is converted to the adenylosuccinate in the presence of an enzyme adenylosuccinate synthetase that requires the GTP as the energy source, and in this step aspartate is used as a nitrogen source. In the next step, the fumarate is released from this adenylosuccinate in the presence of an enzyme adenylosuccinate ligase, and the product is AMP. Now our major product that we were discussing in this topic is covered. Now we will see how the ATP is formed. In the presence of an enzyme adenylate kinase and ATP, AMP is converted to the ADP, and then in the presence of an enzyme ADP kinase, this ADP is converted to the ATP. So this is so simple step. Now we can see that this ATP is then required in the synthesis of the GTP. Now for the synthesis of the GMP from the IMP, IMP is first converted into the xanthosine monophosphate in the presence of an enzyme IMP dehydrogenase. Then this xanthosine monophosphate is converted to the guanosine monophosphate in the presence of an enzyme GMP synthetase, and in this process ATP is used as an energy source which is later converted into the AMP and pyrophosphate. Now for the formation of the GTP, this GMP can be converted into the GDP in the presence of an enzyme guanylate kinase and ATP that is converted to ADP. And further this GDP can be phosphorylated to form the GTP in the presence of an enzyme GDP kinase, and again ATP will be utilized in this process. One point to be noted here was the mycophenolic acid that is the inhibitor of the IMP dehydrogenase, and thus it inhibits this pathway for the formation of the GTP. Mycophenolic acid is a reversible inhibitor of the IMP dehydrogenase. It is an immunosuppressant and it is used to prevent the graft rejection, and it deprives the T and B cells from key components of nucleic acids.